Okay, so define genome. What did you say?
The sum or total of genes in an organism.
So what did you say? Y'all didn't seem to sound very confident about that. What did you say, Janet?
I think the total of the DNA and the organism.
Yeah, so a genome is that you understand the genetic makeup, so the whole genetic makeup total of the genes that make up that particular organism. So define gene. What's a gene?
Recipe traits.
Is a recipe for a trait, which I'll say is like a recipe for a protein to be made, right? Or a characteristic, something that may or may not be expressed. Distinguish between the terms phenotype and genotype. Phenotype is what is expressed or can be seen, and a genotype means what actual molecular genetic makeup is there, right? Okay. Explain the law of complementary base pairing. What did you say? All right, so you gave the examples that it meant that only certain base pairs could pair up together, and it's adenine to thymine and cytosine to guanine. And that's really true. And the significance of that is that then we understand how messenger RNA will be written by that law of complementary base pairing. We know that it's the DNA that has the blueprint of it, but exactly what the messenger RNA will look like. And then, if we understand what a codon is, what is a codon? It's what is it, Brittany? It's a set of 3 base pairs that code for a particular amino acid. That's right. So it's a set of 3 base pairs. And we would know exactly which amino acid would come up and get in that spot, because the anticodon, the transfer RNA, will be holding specifically a particular amino acid. How many different codons are there? 64. And how many different amino acids are there? 20. So we understand that some codons code for the same amino acid. There's codon for starting the chain, and then there's several for stopping the chain. So we kind of get this. Okay, whatever. The 2 steps from getting to protein synthesis, I know my AMP people have had this fairly recently. They are transcription and translation, right? Transcription and translation. What are the monomer units for proteins? And the most important part of building a protein is knowing which amino acid comes up in which space to get lined up, right? And what are the bonds called between them? I don't have this on here, but what are they're peptide bonds that are holding the amino acids together. And which amino acids get in line will dictate that secondary structure, whether it'll be a spiral or an accordion, right? We remember all this? Okay. So good. The monomer units for proteins are amino acids. There are 20 of them. When DNA replicates, makes a copy of itself, each old strand, as it's separating, each old strand is really the template for where the new strands will form on, right? This is called the law of semi-conservation. Very good. So 20 points for being here. I appreciate you all being here. And you had a chance for a bonus because you're reviewing your microbes. Just like in AMP2, you're reviewing what all the time? Hormones all the time. You're going back. Class like this, classes like this, you don't get to just do material and then forget it, put it away, right? You're reviewing the whole time. So in this class, you're reviewing the microbes. What's the etiology of walking pneumonia? The kingdom is bacteria, and the genus? Mycoplasma pneumoniae. Right. So you all are remembering to review these because these can be bonuses for you. How did you do? Did you do okay? Great. Great. That's wonderful. All right. So I am let me just encourage people to do their study guides, because I am going to be using, except for bonus questions, where I'll take microbes, I will be using the study guides to make these quizzes. Okay? Fair? Is that fair? All right. So put the effort in on those study guides and use each other to help you with your notes. making sure your study guides are looking like everybody else's, or that your notes are looking like everybody else's. Does anybody want to bring up anything before we get started on the material this morning? This afternoon, or whatever time it is? Dakota, yeah.
Well, since Sarah and Abigail here don't want to bring it up, they asked me to about the zombie deer virus, whatever it is.
The zombie deer virus. Is that what you're saying? It's a what? The wasting disease. Yeah, that's, I don't know the name of that virus, or if there is a virus that's leading to that. Have y'all heard that? Is it?
It's all over.
Have they actually named it?
The only name I know of is people
Is
calling
the
it.
zombie?
Yeah, but I've seen it being called chronic wasting disease.
Okay, so that's what it's leading to, like a chronic wasting disease. We've seen this before in the deer populations, right?
It's spreading way more now.
Yeah, so it's just, why are they saying, this is really great to be bringing up. I haven't seen this.
They're afraid that it will mutate to where it'll affect
Conjunctive humans.
Because
Well, that's a little
it's
scary.
spreading throughout, the deer population and the animals that eat the dead deer if they have the disease.
Yeah. Right.
And they're afraid that it'll mutate to where.
And are they definitely saying it's a virus? So
It's
Dakota, you
also-
just brought it up and you know nothing.
Yeah, 'cause
Dakota's like, I'm out now.
We
They-
talked about it. It's
They both
also
know
a
my shoulder,
virus.
they were like,
So they do think, so they say it's a prion.
Yeah.
All right, well, if they say it's a prion, if any of y'all eat deer meat, you haven't been paying any attention to anything we've been saying in this class.
We just don't think about it.
If they're saying it's a prion,
They said
Is
it's
it-
similar to mad cow disease.
well, if it's similar to mad cow disease, because you do understand that when you're killing and cutting out a deer, if any drop of cerebrospinal fluid got on any of the meat, right, any of your meat that you were eating, I mean, this is just, that is of concern. That if it were truly you said first a virus though, so which is it? Because y'all understand a virus is not a prion. So is it a virus or is it a
It's a
prion? Did you hear it's a prion too?
prion.
You've seen both? So that means that whoever's writing might not know what they're talking about, because y'all understand a virus is very different than a prion. And if it's a virus, it is true that many viruses are between species. And so we can get viruses from animals, and we can get bacterial and fungal diseases from animals. They're called zoonotic diseases. Y'all have those in your notes somewhere. And we also know that viruses can evolve and then jump species. So never before been seen in humans, but all of a sudden now can, because they've evolved. Viruses can. But if it's a prion disease that's of the central nervous system, and if that's really true, y'all listen out to see if that's really true. And if that really is true, you should not be eating deer meat. Do you think I'm being an alarmist? I'm not being an alarmist. Do your research on prion diseases. Is there any cure? They're terminal. It's over.
I think they said right now it's in like 25 states and plus Canada.
Okay,
So it's-
who is they and where were
I've seen it on news
Okay,
websites
so y'all need
and
to,
people sharing it on social media.
okay, social media is not the site to get news,
No, I'm
ever.
talking about I've
Not
seen it
even
on
ever.
news websites.
But I'm just saying, because it isn't, because they'll create things for sensationalism so that you click on it, and they're clickbaits, aren't they? Social media news. So unless you see and even when you see NPR or wherever, you have to it's still clickbait. Go back out to your Google, do the search. I appreciate you guys bringing this up. This would be a huge concern for those of us in rural America that love deer meat. Who loves deer meat? A lot of people, right? The rest of you don't eat deer meat? Are you vegetarians, the rest of you? Okay, just don't like deer meat. Okay, but anything that is of huge concern. It really is. Thank you for bringing it up. You win, so mine doesn't seem very important now.
Thanks, Dakota.
But what we need to do is to make sure that our information's legitimate and look to see what the real numbers are, see what this is really about. I have seen in an article that from supposedly a good site where they'll talk about an infectious disease and they'll name it, they'll call it first a virus, then they'll call it a prion. I've seen Sanjay Gupta, y'all know who he is? I've seen him talk about something as a viral infection and then call it a bacteria. Do you get what I'm saying? Would you all ever do that? You all would never do that, would you? Because you know they're very different, right? I love Sanjay, but I'm just saying. I don't know him, but I like him. He's mighty handsome. So anyway, I'm just saying, but he is. But people will get that confused. So we need to be looking at that. If they have said that it is of the prion class of infectious agents, because are prions alive? No. But if they have said that, then we know that can be. That's why we're very careful now with our meat, you know, and people have to be very careful of this. There is no cure for that. We're going to see today, we're going to talk about prions again, and they're the most difficult thing to get rid of. The most difficult thing to get rid of. As far as like when you're cleaning or disinfecting or trying to do whatever, prions are the hardest. So, okay. Does anybody have any other questions that I couldn't think of to ask Abby and Sarah? Have any of y'all seen that? Yes. Y'all had seen it too? Where have I been?
It's all over social media.
I've seen it on a couple.
I've seen it on
sites,
like
too.
one news show, and it was like one time.
Okay, so we need to look that up, because guess what? It might be just because that would be rural America that's affected by that. Who cares about rural America except for us in rural America, right? We care. What'd you say?
I said I had lots of people in my fridge. Yeah.
Yeah, it's one of the reasons I stopped eating like that kind of meat, even from people who I knew where they were hunting and what they were hunting, and that they were good hunters, so they knew how to cut up the meat. They knew how to do. It's one of the reasons I don't eat wild game anymore. It's for this very same thing, because we just don't know. He doesn't want to believe it. Well, we might not want to believe something, but we can't be like, stick our head in the sand. Okay, so guys, I have a couple of things that I saw. Does anybody else have anything that's really good to bring up? Does anybody else have anything in the news? I think there was a salmonella outbreak. Did y'all see that? Okay, I didn't remember enough about it right now to actually. It was 20 pigs back there being quiet, but I know I was watching it when he was seeing. I saw it when he was looking too. It was like 25 states or something. But anyway, so that's something to be careful of, and I don't know, I didn't keep enough of the information in my head long enough to tell you all about it, so just maybe. What'd you say?
Like during like mid-December, there was like something about deer having like tuberculosis, and whenever you scan it, you like see like the spots in their lungs.
You weren't supposed to eat them. Yeah. Like I said, be careful eating wild.
Yeah, it's being recommended to get the meat tested before.
Yeah. Wow. I hadn't heard that either. So, yeah. Okay, we're going to talk about some parasites, some worms you can also get. So that's actually very common, and not always are they macroscopic that you can see a gross anatomy when you're cutting up the meat. Sometimes they're small enough that you can't really see it. So, but you could still get the worms yourself. They could move to your own muscles. Okay. Is anybody hungry? All right, here we go. So the this is from UVA research, and it made the top page of the health thing. I was really happy. Richmond Times, it actually is making national news right now. But there's an antidepressant that could be a sepsis treatment. Does everybody remember what sepsis is? What is sepsis? Because you would need to know that. Yes, well, it's not just bacterial though. It can be any microorganism. So it's microorganisms growing in the bloodstream, actively growing in the bloodstream, right? And it's usually bacteria, but it's not always bacteria. Sometimes it can be fungal, can be other things, but that's sepsis. And you know how I stand up here and I tell you all a fourth of the people who have sepsis that have medical treatment will die. And I say that because that's really, and it would depend on it would really depend on which microbe was causing the sepsis, because some of them have a much higher mortality rate than that. Some of them have a little bit lower mortality rate, obviously how virulent the organism is, right? So I just think that's horrible, because you're thinking about these 4 young people, and it doesn't matter age, doesn't matter whether they're immunocompromised. You know, like we kind of tend to think in infectious disease that 1 in 4 are going to die with the best medical treatment. Best medical treatment. This article says 1 in 3. So I don't know if the numbers are actually changing. I would want to look this up if the numbers are changing or not. But is sepsis serious? So sepsis is very serious. But what these researchers found when they were looking, because their focus is to take drugs that have had tons of clinical application, meaning that they have a lot of people that they followed over many years, because this antidepressant's been out on the market a long time. So they can take these people who've been on this drug and do retrospective studies as far as like the side effects of this drug. So this group is looking for drugs that have been on the market for a long time, no matter what they've been on the market for, no matter what they've been used to treat, and seeing if there can be something else that might be good for them to be treating. And what they did was they took this antidepressant, which is actually used for OCD, that types of behavior, to treat obsessive compulsive disorder. And they're looking to see if it could influence a protein called sigma-1. And this sigma-1 is a protein that's often, this receptor is often not functioning if somebody has sepsis. And so the sepsis itself ends up causing, the inflammatory response causes so much damage to the person that this is why the people die. You know, a lot of times when people have sepsis, you can give an antibiotic or an antifungal and you can get rid of the organism, but it's the person's own inflammatory responses that do so much widespread damage that the person's sometimes going to die anyway, right? So looking to see if there's a way to inhibit the inflammatory response. And this particular antidepressant for OCD has having an amazing positive results in the labs, and they've already started doing it. I was trying to see if I had highlighted that, but it's like reducing it by at least a third. You know, that's a lot, right? So that's actually, no, wait a minute. It's reducing it by more than that. And I can't find the actual statistics right here, but y'all can come read this. So, compared to 62% of untreated mice. Oh, only 9% of the laboratory animals died compared to 62% of the ones that didn't get it. So that's an amazing difference, right? So anyway, I think that's going to be something for us to be looking at. And then another article, anybody have any questions about that? I think that's really kind of a way to think outside of the box. Look at some of these, because we know we have lots of chemotherapeutics on the market that we've been using for a lot of things for a long time. So instead of trying to create new things, maybe looking at some of these that we know are relatively safe, you know, because they've been used for so long, seeing if there's something else that we could pinpoint treatments for. So I thought that was really kind of ingenious. These researchers that are doing this is what they do. But the other thing that's in here is an inexpensive, simple way cleanup routine wards off superbug infections. You all know that a risk of going into a hospital having any kind of procedure done is having a superbug infection, which is one of the most common ones. Yeah, so the MRSA. So Brittany said it out. So methicillin-resistant Staphylococcus aureus, so the MRSA. And we understand that much of the time that actually comes from the patient themselves. And then the patient goes home and they end up getting a secondary infection that ends up being worse than what they were in the hospital for the first time, right? And they can also be spreading it to not just family members, but to the community. So now we talk about hospital-acquired MRSA versus community-acquired MRSA. I think that's splitting hairs, but because who cares where you acquired it? You've got it living up your nose. After the break, spring break next week, you guys, when we come back that Monday, you all will be doing your own cultures of your own couple of different body parts. And if anybody works in a skilled care facility or whatever, you can do whatever body part you want, but I would hope you would do your nose. because you'll be able to see whether you carry Staph aureus or not. And if it is Staph aureus, that's enough that it grows on those plates, it is likely to be MRSA. So, because most of us do that have ever worked in any kind of clinical setting. But this is a little procedure that they're doing, which sounds kind of like it wouldn't be fun, but maybe you get used to doing it. They're sending home a medicated goo that the patients for the first, this sounds like a long time, 6 months. That's a long time. Who's going to do something for 6 months? But for 6 months, they would daily swab their noses with this, and they would use a special kind of hand cleanser. Now, they didn't really talk about what the hand cleanser is, but I think it's probably like a Visahex that you can get just at Rite Aid or whatever. And this is absolutely, it looks like it's getting rid of this. So they're no longer carriers after that time. So that seems pretty good, right? If somebody would do that, if somebody would adhere to that type of hygiene treatment afterwards to reduce their risk of carrying and or causing more infection. Do you think people will do that? You would? For 6 months? Yeah. I guess it depends on how gooey the nasal stuff is, right? But I know it didn't sound very attractive. Okay, so what? Like at night, put this goo in your nose, right? So can you breathe? But anyway, I don't think that would be a bad thing to do. All right, so are y'all good? Do you have any questions for me? Should we keep going? Somebody is going to help me to know where we stopped. Where did we stop? I have no idea. Oh, so we were talking about mutations. Are all mutations bad? Did we stop there, or where did we stop? Will somebody let me know? Oh, we had not started mutations.
We stopped with
Oh, we had.
the messenger RNA,
Okay.
all that.
Oh, yeah. Okay. I'm with you.
We did codon and anti-codon peptide.
Okay. Oh, so we're not going to have lab today. On Wednesday, I'm going to get you all to pour media. So you're going to be making some plates on Wednesday before you leave. And then some of those plates that you make, I'm going to put them away the next day. And y'all will use some of those plates when you get back to the group. But we'll do that Wednesday, okay, that you make plates. All right. So we were really just ending with this idea that you understand that it's your nucleic acids, it's the DNA that dictates the traits you have. And what's being expressed is what we call the phenotype, what information, all the information itself is called the genotype. And we know how you get from DNA to proteins being made. We know that cells are essentially just protein factories. They can make lipids as well, but we understand that you get there by transcription to translation, and we understand those processes. Were there any questions about that? No? So we're pretty good about that? And we know what the genetic code is. What the genetic code means that you know for each codon, each set of 3 bases on the messenger RNA, exactly what amino acid would be called up at that site. So there's some control over that, though. And one of the things that I wanted you to know about the control is, and let's see if I can find that slide. I might not be able to, but anyway, there's control of that because you don't need everything happening at all the same time. So maybe that would make sense to you. So bacteria have this that's referred to as an operon. I'm not going to ask you about it. It really isn't. But what it means is think about bacteria. Just think about the bacteria in your colon, in your gut. Okay? Many of the bacteria, one of the predominant bacteria in our colon is E. coli, Escherichia coli, and all of our colon. But let's think about this. And bacteria have the ability to use lactose. What is lactose? No, it's the milk sugar. That's exactly what it is. And so anyway, can have the ability to use it sometimes in their metabolic processes. But would they need to have that ability happening all the time or only when milk was present? only when milk is present. Wouldn't that make sense? So they can actually, from their environment, know when they need certain things, and they have this mechanism where they can turn these genes on and off. So when you talk about these regulators and control of what's being expressed, that's one of the things this is talking about. Okay, so we get that, and that seems like that would be a very efficient way for not just bacteria, but even our own cells. Even our own cells to actually work. This seems like this is out of order, these slides. But okay, I'm going to try to do it from the order that it's in. So we do know that DNA does not have sexual reproduction, does it? DNA. Bacteria don't sexually reproduce, do they? They just clone themselves. So this is a type of just making a copy of themselves. The DNA will make it replicate itself. The cell will double in size. It will split down the middle. 2 new copies of each of the old copy of DNA will be in each of the 2 new. And that's really like a clone. When we think about recombinant DNA, this is any organism that contains genes from another, that originated from another organism. So when we talk about recombinant DNA technology, it means a way that we can actually take DNA from one species and put it into another species or into another organism. We've talked about plasmids. We know that they can be transferred, can't they? They can be transferred through little pili. We know that they can do that. And sometimes those plasmids have traits that we care about. They're traits that can make an organism more resistant or can give an organism properties that can make them more virulent. So we know that plasmids are one of the ways, but there's also sometimes these chromosomal fragments, and these fragments can also be picked up by other organisms in the environment. So there's 3 modes of how genes can be transferred in bacteria: conjugation, transformation, and transduction. So we say conjugation is actually where they're using the plasmids and they're sending this little pieces of DNA to another cell. Transformation,
Bacteria.
yes, bacteria. The transformation is this ability, and I want to give you an example of what goes on here. But this is actually a live free donor, so fragments of donor DNA into a recipient cell. Let me give you an example for transformation. This was discovered, and I know they've already told you this story, but this was discovered that this can happen, and it can happen even when these pieces of the DNA are actually out in the environment and be picked up by other species. So let me give you this example. They were looking at Streptococcus pneumoniae. So some researchers were looking at Streptococcus pneumoniae, and most of us have some of that even in our throats, Streptococcus pneumoniae. Is everybody good?
Yeah, I have a slide.
Okay. So Streptococcus pneumoniae is a bacterium, and obviously we know it's associated with what disease? Pneumonia. Not strep throat, because that's something different, right? Mm-hmm. But pneumonia, and that's why this was actually called the species, because we know it causes a really deadly bacterial pneumonia, has a really high mortality rate. But it also can cause conjunctivitis, and this isn't your list, so you might want to make sure you know this organism now. Streptococcus pneumoniae. It can cause conjunctivitis, which people call pink eye. Certainly causes pneumonia. It can cause meningitis. It's probably the most common cause of bacterial meningitis in this country right now. Bacterial meningitis. And it can cause middle ear infections. So is this organism, do you think this microorganism is pretty much everywhere in the environment? Yeah, it is. And most of us, many of us have this in our throats. Okay, but we don't have the serotype that is so virulent, so deadly. So let me say this. They've taken 2 strains of Streptococcus pneumoniae, one that had a capsule, one that had a capsular, that was enclosed in a capsule, and one that didn't. And you can actually see macroscopically, you can see the difference of them on a plate, an agar plate. Strep pneumo that's encapsulated is glistening and it looks wet. It's got a really wet looking little colony, where the ones that aren't encapsulated are really dry. And they have like a little umbilical thing in them. Some of you all will see that you have strep, but you don't have the one that's encapsulated. Okay? So, anyway. But what they did was they took strep pneumonia. Did I tell you the story yet? This is important for transformation because this is making you understand what transformation really is. So they took Streptococcus pneumoniae that was encapsulated, that had this capsule, and they injected it into a certain number of mice. And they took Streptococcus pneumoniae that didn't have a capsule and injected those into mice. Guess what happened to all the mice that got the encapsulated strep? They all died. The ones that didn't have the capsules, none of them died, right? So then they thought, okay, well, what would happen if we took, I did tell you all the story, did I? Okay. Well, let's see if y'all can figure out what happens. Okay, so then they took the encapsulated strep pneumo, and we know it's virulent, right? It killed every one of the mice that got it, caused them to get very, very sick, septic, died. They took it though, and before they injected it, they heat killed it. They heat killed it. And then they injected it into the mice, and guess what happened? They lived. All right, so the moral of that, you would think most people would stop doing what they were doing after that. They think, okay, well, make sure you got something, maybe heat will kill it, right? Okay, well, wait a minute. Griffith was this researcher, but anyway, he said, well, what would happen if I took the ones that were killed? They're dead. They didn't kill a single mouse. But what would happen if I took that microbe and I mixed it with the ones that were harmless? Okay, wait a minute. The ones that were killed didn't kill anything. The ones that were harmless didn't kill anything. But guess what happened when they mixed the two together? 100% died. What had happened? The bad bacteria were dead. They were heat killed. We knew they were dead. We knew they couldn't do anything because we'd injected the mice and it hadn't done anything to the mice. But what happened when the heat killed one were put with the friendly ones? The friendly ones turned what? Evil. The friendly ones, guess what the friendly ones did?
Transformed.
They transformed. And they all became what? They all became evil. Do you understand that most people wouldn't have thought to keep going after that? They'd have thought, okay, if you take a pathogen and you heat kill it, it's not going to hurt anyone anymore. Except that we still continue to see sometimes these encapsulated streps showing up. So what happens is what we know can happen is that when the heat killed organisms released their cells ruptured. What was in the media? Pieces of DNA. Some of it that encoded for a what? For the virulence factors that were going to end up being taken up by the friendly strep. And then the friendly, nice little strep transformed into what? Evil, wicked. Okay, it sounds like a fairy tale. But that actually happens. And so do you think it just happened? Here's the moral to that story. Do you think it just happens in test tubes? Do you think it just happens in test tubes? No, it can happen in our own microbiomes, can it? It can happen in our own microbiome. So we know that this is happening. But can we actually use it? Can we actually use this to our advantage maybe? And understanding that this is the way sometimes we can use recombinant DNA technology to actually make something instead of wicked, make it, just change it in a different way that will be more beneficial. Yeah, and this is actually done. So anyway, and then there's something called transduction. So this is using bacteriophages. Do we remember bacteriophages? What are bacteriophages? Viruses that infect what? Bacteria. And this is a way that we can actually use the bacteriophage as a vector to introduce pieces of DNA that are encoding for certain traits into other organisms. And this is called recombinant. Using this, we know this happens. We know this happens in nature. All three of these things are happening in nature, but can we use these things to actually also in recombinant DNA technology? Yeah. Did that make sense? Okay. Did anybody have any questions about that? So anyway, this shows you a little bit. They show you pictures of this, and I'm not going to talk about transformations. I know y'all talked about that, and I know you talked about that in regular biology. Okay, so here's another thing that we think about. Anything that would change DNA, anything that could change DNA, whether it is conjugation, transformation, or transduction, any of these things, and even naturally occurring changes in the DNA, spontaneous changes in the DNA, can lead to a genome mutation, which means that the sequences of the bases on the DNA template have changed. For whatever reason, they have changed. And if they have changed, sometimes it's just a benign change, we said. It's going to code for the same amino acid. And if the change happens and it codes for the same amino acid at that site, would there have been that's a benign mutation, right? It doesn't change the protein. It's not going to change anything. Sometimes it changes things and it's actually adaptive. So it's an adaptive trait. It's a trait that can actually help you to survive. And then sometimes the mutations are immediately lethal, or sometimes the mutations are simply just bad and it's not going to be lethal immediately, but it can cause problems. Yes?
You said if they change and they have the same amino acid, what are they called?
Benign. Okay. Just benign, meaning it's harmless, kind of. It doesn't really change. So a wild type, when you hear about a wild type, that's a microorganism that's natural, non-mutated characteristics. So that's what's referred to as the wild type.
Thank you.
So people are writing things down verbatim. Does this make sense to you? Remember I drew out a DNA recipe strand? And I told you that the nucleotide sequence dictated what was going to be written. What's going to be written from a DNA strand? What's the strand that's going to be written? A messenger RNA. And the messenger RNA is written in a very specific way because of the law of, because of the base parent. But if at the DNA, if the DNA strand somehow got changed, got transformed, or pieces of other DNA got included in that, could that change the messenger RNA? And if it changed the messenger, that means it changed the codons. And if it changed the codons, what did it change? The sequence of amino acids. And does that make sense to you, Brandy? Mm-hmm. Okay. All right. So any change, that's right. When you talk about a wild type, that's the normal. That's what you see. That's what's in the wild. What's in the wild we think of as being normal, right? A mutant strand is going to show a change from that, though. And can that change the morphology? Certainly it can. Can it change nutritional characteristics? Can it change resistance to chemicals? Of course it can. It can change a lot of different enzymatic functions and just what they produce. So yes, it can. These are the mutant strains. Spontaneous mutations are those that we think of that we know happens in every species we've ever studied. Every living species that's ever been studied, there is a known rate of spontaneous mutation. Those spontaneous mutations are rare, but they do happen in every species. And when we think about the spontaneous mutations, they're typically going to be like inversions. So you would have something like, and this would make sense, it's called an inversion. So maybe you have an A, T, T, C, G, right? And in an inversion, when this was being copied, one of the strands, these two would have been inverted. And if they got inverted, what would be changed if this was a sense strand for a recipe? The messenger RNA. And could it potentially affect the sequence of amino acids? Yes. So we get this. So it can be an inversion. It can be what's called a deletion. So something could be deleted. A nucleotide could be deleted. What's a nucleotide? I could have asked you that. What's a nucleotide? It's a phosphate, a sugar, and an amino. And this is one nucleotide, and another, and another. Right? So there can be a deletion. There can be inversions. There can be substitutions. These are all things that we know can happen at this DNA level, and they do happen, but it's really, really rare. But that rarity is enough that it's given the diversity within a species. That's what gives diversity within a species. Induced mutations can be encouraged by using like chemicals and radiation. So you can actually try to induce mutations. Why would you ever try to do that? Well, they're kind of not doing it for so much for that, but they're doing induced mutations to try to stop microbial growth. Because you can induce mutations that with UV light and X-rays and whatever, that can actually reduce the number of microbes at a certain site. Have you all ever heard of using UV light to reduce the microbial numbers on food and whatnot? Sometimes even using radiation. Have you ever heard of that? So did you know that some of your water and some of your food goes through radiation to reduce the number of microbes? Does our food need to be sterile? No, it doesn't, but much of our food is coming from long distances away and is being stored for long times. What does that encourage the growth of? Microbes. So what's one of the ways that we can do to try to control microbial growth? Yeah, some of this. So great. So this is what I was talking about, these addition, deletion, substitutions. You know, those are just some examples. So those are just some of the examples that I had mentioned. Some of them, as I said, and I don't care about this stuff, but some of them I said are going to be benign. Some of them are going to be what? Adaptive traits, just going to give variations of traits. Some of them will be lethal or not so great, right? We have a lot of built-in mechanisms in the DNA to prevent, that are proofreading, that are preventing these changes, and this is why we see them happening so infrequently. But this talks about some of the positive and negative effects of mutations, so it goes to some of that. Now, this is what I want. This is what I want you guys to this is a couple of things I do want you all to know. So I would make it red here so that you guys can see that this is something that I do think is important. If it ends in ASE, what is it? It's an enzyme. There are these enzymes known as restriction endonucleases. These are enzymes that have the ability, these enzymes, they're actually seeking out and have the ability, even on foreign DNA, of breaking the DNA at a particular site. At a particular site. So what this means is that you have these enzymes that can be, that we actually know about, and we have, that when they're included in DNA, they're only going to cut the DNA at particular nucleotide sequences, like where there's a T and a C, they'll cut it right there. Or they'll cut it at a G and a C. You get what I mean? They'll cut it there. So we know exactly, they're almost like scissors, if you will. So what you might be thinking is, so what? You know, so what do we do with this? So this is how biotechnologists actually cut DNA at very specific points of where they want it cut, how they can cut out certain recipes for certain traits at very particular points. So this is absolutely necessary, this technology of having these restriction endonucleases, having these enzymes available for recombinant DNA technology. I had already mentioned to you guys that they can take the human DNA and cut out the gene for human insulin human clotting factors and insert those into other microbes using recombinant DNA technology and get those microorganisms to start producing human insulin or start producing human growth hormone or start producing human blood clotting factors. How do they do that? By knowing exactly how to cut the DNA, where they need it cut, and then isolating that recipe out. This is used for that. These enzymes, this is all I want you to get from this, is that these are enzymes that are used for that. By the way, these enzymes come from bacteria. So, because bacteria have these enzymes to protect themselves, they use these enzymes to protect themselves. But what biochemists have discovered is that certain bacteria produce certain scissors that will cut at specific sites. That's what these are. Is that making sense? So Virginia Tech is one of the leaders in this, was one of the earlier leaders in this, and they actually have an entire library of microbes that produce specific enzymes that they know exactly where to cut. So if somebody wants to cut DNA in a particular sequence, they can call up, not making this sort of whatever, but they can get someplace like Virginia Tech, and other universities have it too, but they can say, I need this enzyme that will cut here and here, and just right at those particular sites, and then they get them. So this is what's needed for that. All right. All right. Let me see what else I wanted you to get from this. All right. So this, again, this is something else I actually wanted you to know about. These are called restriction fragment link polymorphisms. Oh my goodness. This is why they call it RFLPs. Restriction fragment link polymorphisms. This is, again, every species has these known RFLPs, these known ones, and how there's a difference in the cutting patterns of these endonucleases. So every species is unique in these RFLPs. And so anyway, that's kind of all I wanted you to know about that, is that they're unique. Each species, each enzyme, this is going to be kind of unique. All right. So reverse transcriptase, what do you know it is? What is reverse transcriptase? It's a what? It's an enzyme. And in reverse transcriptases, these enzymes help to go from RNA to DNA. Wait a minute. We didn't go, that's not how we go. We go from DNA to messenger RNA, didn't we? But this is how we can use these enzymes to go from RNA back to the DNA. But how would we know that would be possible anyway? Because of the law of what? Complementary base pairing. And it takes these enzymes to do it. So this is where I'm going to bring in viruses now. Viruses are not considered alive, are they? They're considered active or inactive. But when they're inside of a host cell, can they be active? And to be active, if it's an RNA virus, what would it have to first do? What do you think? RNA viruses has to have this enzyme to be able to clone back to and it's really cloning the DNA to go back to the DNA. So if we know this, if we know that these viruses, these RNA viruses, which are some of the most dangerous viruses we know, HIV and rabies, right? What might we target as far as treatment? That enzyme. Because is it going to take this enzyme for that virus to replicate itself? Yes. So this is an enzyme. What I want you to get from this is that this is an enzyme, reverse transcriptase, that is an enzyme that helps to go backward from the RNA to a DNA molecule. And would that essentially make a copy? You could say little c DNA, is that a copy or a clone of the DNA? Wouldn't that essentially do that? Guys, would it? Complementary, copy, clone, it can be made for that. Exactly. Now, I don't have the gels. I used to have people do gel electrophoresis for DNA. But what I want you to realize is that when they're looking at DNA, they're looking at these pieces of DNA that have actually been treated. They're using a technique called gel electrophoresis. And when we think about this, we can look at the text of it, but when we think about this gel, you know it's getting cut. Are we using the endonucleases to know where to cut? Or do we know which ones to use depending on what we want? Yeah, so different, and there's hundreds of these that have known cutting points, right? So depending on where you want to cut. Now, then you have a sample, and here's what I want you to see. This, and I'm going to explain this to you. This is an auger that's see-through. So y'all all know what the blood auger looks like, and you knew what chocolate looked like. Well, this is auger that's actually see-through. So you can actually really, it's gel and it's semi-solid, and you can see through it. And there are these little wells here at one end, these little wells, and these wells are going to be cut out of the auger so you can put your specimen in the wells. Does that make sense? And these are 5, well, this is one with known markers, but there are 4 samples here with one that you know what the marker is. So in these little empty wells, you have put specimen. Now what you've then done is actually put it into an electrical field. And so after you put your unknown samples and your known sample, you put it into an electrical field where there's a current running through this auger. So what ends up happening is, because of what attracts? Opposite charges, right, attract. So these molecules of DNA are going to be being pulled through the auger to the opposite end. Obviously, would it make sense to you that these heavy ones can't move as fast as these little lighter ones? Do you see this? So they can't move as fast, and they won't go as far as some of these lighter chains. But you can see that it's pulling through. Now, the known marker would be, you would know exactly which DNA you have at these points. But then on your unknowns, you see how they look different? All these unknowns look different. So they can tell you what sample 2, what type of DNA it had there, which one of these it was, right? It was probably one of these lighter ones, these littler ones, right? Restriction fragments. And then they can go in there and get those restriction fragments because they know that's where it is at that particular time after it's been incubated. Does that make sense? Does it? Yes.
Who's figured out how to do this?
I know, I agree. Okay, so its purpose is because then, okay, that's a great question. Who's the first one that started this? You know, I don't even know who some of the earlier pioneers in this were, some of the names. But you could go back and look. This has been available for decades, though. This has been happening for a long time. If you've ever heard of, here's one of the applications. I'm going to give you a couple of applications. Some of the applications is that then we would know where that recipe is. We can pull it out right there and then put it in recombinant DNA technology, put it in another species, right? And get them to do that. If we could get them to take it up, you know, and there are different ways that you can get them to take it up. But let me tell you another application. Human beings. Each one of us has a DNA fingerprint. Y'all have heard of that, haven't you? That we all have a DNA fingerprint. This is how they do DNA fingerprinting. They would take, let's say this known marker here, this little well was actually a suspect in a crime. And what they do is they expose human DNA. When they're doing this, they have known RFLPs. We all have our own unique ones. We all have our own unique repeating things, every one of us. Nobody's the same except for identical twins. Identical twins have the same DNA fingerprint. They don't have the same actual fingerprints, but they have the same DNA fingerprints. But if you don't have an identical twin, nobody else has your same DNA fingerprint. Nobody. So they would take a suspect's DNA, they would put it in this well, and they would see where the bands were. Right? And then they would take people who they've gotten DNA from, to rule them out as suspects, and they would put them in these other wells. What would happen if one of these people they're trying to rule out had exactly the same banding as their suspect? There's your person. There they are. That's exactly right. And that's how they do that. Couldn't it be somebody else? No, it was OJ. It was. It's just that they didn't know, they didn't understand DNA. I'm just saying, they didn't understand DNA technology at the time, and nobody could explain it to the jury that that was that specific. Are they also not just doing it they're not just doing that to get criminals, but they're also doing this to get people off, aren't they? Are they not? So yeah, so this is actually being used for that as well. But it's also being done to isolate pieces of DNA that we want. So if we wanted this piece, and I don't know what this particular one was talking about, but if we wanted this particular piece, would we know exactly where it was, and could we be able to isolate it from that?
So what's like an active definition?
Gel electrophoresis is a way of cutting up the DNA and then isolating certain components of that DNA. Because guys, is DNA really long? Yeah, it's really long. Now, what's it going to take to do this? You're going to have scissors. What are the scissors that cut it? Yes, the endonucleases, right? The restriction endonucleases. Do we have specific enzymes to cut specific pieces? Yes. Yes, we do. Does that make sense? That we have specific enzymes to cut specific pieces. Now look, how do you ever get, and this is going to be important kind of for you to know too, how do you ever get enough DNA that you could actually see? Because like think about a bacterial cell. We said our cells are huge old cells, and we have a ton of DNA, don't we? 23 pairs of chromosomes, don't we? So we have a ton of DNA. But what about little bacteria? Like how would you ever get enough of even the little recipe that you wanted, or whatever you might be doing? How would you ever get enough of, let's say you had one human cell left at a crime scene, only one was left. Let's say there was a hair fragment. Did y'all just see where they just got some guy who committed a horrific crime like 30, 40 years ago? I think it was a little 11-year-old girl disappeared. Did you see this? He threw a napkin in the trash can, right? First of all, what man uses a napkin? No, that sounded so sexist, didn't it? No, I'm just kidding. I was joking. Okay, I wanted to see if y'all were listening. Wake up. That was just, but he threw a napkin in, and he had enough. What are you talking about? You could take a napkin and you could do something like this and get his DNA fingerprint out.
You replicate it.
How?
Science.
Science. You use PCR, polymerase chain reaction, as a way that we can replicate. So you can take even just from one cell. You didn't even need all the cells that he wiped on that napkin. There were plenty there. You can leave one cell. Start with one cell today, and after 20 cycles in a PCR, you should have 1 million copies. Will they be identical to the first? Why? Because of the law of complementary base pairing. PCR is called polymerase chain reaction. It is a way that we can rapidly increase the amount of DNA that we're working with in a sample by replicating it, making copies of it. And it takes these repeating heating, cooling cycles. It only takes about 10, 15 minutes, heating, cooling cycles, where you heat the DNA. And what will DNA do if you heat it? Why? Well, they're not peptide bonds. It's weak hydrogen bonds holding the strands together. So you heat it just enough that it separates. Then as you're cooling it, you've thrown in all kinds of nucleotides, right? So new strands form. You heat those 2 new strands, they separate. Continue throwing in your stuff so that they replicate themselves. And then you can have, and it talks to you about the numbers that you can have after just a very short cycling time. So now this technology has gotten so efficient that they can do DNA fingerprinting now in a really quick hurry. It doesn't take long at all. It used to take so long. but it doesn't take long now at all, and it's not expensive now. Yeah.
So sometimes maybe common 40 years ago, and they still have like the evidence, but didn't have the technology like process it. Would the evidence still be able to go?
Yes. Yeah. And they're doing. Have y'all been hearing that they've been like people have been getting out of jail because they've been locked up for decades, but when the evidence is done, they realize that they had the wrong person in. Have you been hearing this? This has been happening, and it's because of what you said.
It just
Yeah.
sucks.
It just would be horrible to think that somebody, but you know, it doesn't. You know, it doesn't happen often. But if it happened to you, would you care? Yeah, pushing. It doesn't happen often, but it happens enough. It happens enough that people should be, you know, very much needing to want to know whether, you know, might actually want to make people think about the death penalty too, you know, because if without solid, you know, whatever, because there have been people who've been on death row within hours of being executed. And then they find out, you know, the execution was staged, but then they'll find out they didn't even do the crime in the first place. They got that close to being executed. Y'all can look up those cases. They do happen. So PCR, I want you all to know that Carrie Mullis, I know I've probably told you this in another place, but anyway, Carrie Mullis with a K, K-A-R-Y, Mullis, M-U-L-L-I-S. Carrie Mullis gave us this technology. I do know his name, Jared, and he won a Nobel Prize for the PCR, coming up with PCR, which is a way to amplify or get more DNA in a quick hurry, right? So Carrie Mullis came up with this. He talked about how he thought about it driving while he was high down an interstate in LA, in California. It was the name of the book he wrote was Driving Naked in a Minefield. And yeah, he was very eccentric, but anyway, he was driving home, and what he thought about was, you know, how frustrating it is that they, you know, how long it took and how much of a sample you'd have to do any sort of testing of DNA. And thought there's got to be a way that we can just make more of it. Like there's got to be a way. It's there. How can we make it? And he started thinking about what law and how DNA molecules held together, and how if you just put it through this heat, it wasn't as simple as I'm talking about, but putting it in it took him a little bit longer. But that's how he thought about it. He thought about it driving home one day. He really did. And then he went back and started working on this technique. And now the technique is used everywhere. Every single place this technique is used. This is used in some, you know, almost every hospital biology lab department. This PCR machines you have instruments there that are amplifying this. They can use it for DNA probes. So now instead of, and this is where microbiology is kind of going. We said that the catch-22 is that we have to grow these organisms through biochemical testing, which you all are going to do. You have to do stains and biochemical testing, and then get to your ID and send it to your patient. And by that time, could the patient be dead? Right. So, but what would happen if you had a probe for a particular organism and you knew exactly what this organism had, this unique DNA type of sequence? Could you probe to see if your unknown had it? Could. And they used this PCR for that too, for DNA probing and that type of thing. And those results are pretty immediate. Instead of growing the organism, staining the organism, doing biochemical testing on the organism. Yeah. So anyway, so we've got these bacteria now through recombinant DNA technology for doing this. We can actually mass produce hormones, enzymes, vaccines. These are some of the applications for getting those things out. But it's also for some of the forensic science too that we were talking about. All right.
What is the genetically engineered bacteria?
Yeah. So getting, you know, because I had talked to you guys about that, have I not? And we said we do get it, that we still can use plasmids, we can use bacteriophages. That's what's being used in some of these technologies. All right. So I do want to start on this because I'd like to finish this chapter, chapter 9, and I want to give you all a little heads up here. So chapter 9, this is really talking about controlling microbes. We live in a microbial world. We kind of want to control the growth of microorganisms, especially if we have somebody who's at risk, right? So, or if we're about to operate or do something invasive to somebody that we certainly want to be careful of that. So this whole chapter is this, and most of this chapter is going to be definitions, you'll see. So I am going to kind of roll through some of these definitions because I think that, you know, a lot of you have heard this terminology before. So most of you have heard what sterilization, disinfection, decontamination, and antisepsis, haven't you? You've heard those terms before. But I really kind of want you to just have like working definitions of these. So this little concepts in these, you can see some of the working definitions and some of the key points. So is it possible to sterilize a person's skin?
Not completely.
No, it would not be possible to sterilize. And when we say sterilize, we really want to realize that if you're talking about sterilization, you're talking about destroying or removing all the microorganisms, including viruses. You're removing everything, right? You're talking about a sterile environment. Okay? Now, when you, pardon?
What is the orange stuff?
The iodine? Yeah, the iodine, it can greatly, that can actually, it's pretty toxic and it can take the numbers down to lower than just alcohol. So you do try to, if you're doing, let me, okay, so I'll just talk about this now. So we know we can't sterilize skin, but if we're going to do something invasive, like draw somebody's blood, wouldn't we want to knock the numbers down a little bit before we stick a needle through their skin, into their bloodstream, their veins, right? You would like to do that. So in a typical blood draw, phlebotomy, you are just going to alcohol off the area pretty well, right? Or if you do a finger stick, you're going to alcohol off and just a little bit, try to get that numbers knocked down a little bit. Because even though bacteria might be being introduced or microbes might be being introduced, they're not going to stay there long because you have cells that are looking for them and they're going to take them out, right? But what if you needed to do blood cultures? So you were taking somebody's blood and you had to culture it. How can you culture somebody's blood when you know that it's probably going to have what kind of contamination? Skin flora, right? Skin flora. So instead, you can't just use alcohol. You're going to end up treating it differently. You're going to do iodine. You're going to let it sit for a few minutes. You know, you're going to do this. You're going to go through this procedure that you know is going to greatly reduce it. Same thing is if you're going to do an IV. Because if you're going to put in an IV and you know that this is going to be something that's going to have a direct route, wouldn't you like to kind of clean that area a little bit more than a regular phlebotomy would be cleaned? Right. But can you actually sterilize someone's skin? No, you can't. Now you talk about disinfection. And when we talk about disinfection, is this going to be sterilization? No. But again, it's usually on inanimate objects. So to disinfect something, this is what we do when we're thinking about trying to clean our countertops in the lab, right? Disinfection. We all agree with that. But that's what we're trying to do. So you're looking at things like bleach, not iodine, but anyway, but you can use these different things for disinfecting. Decontamination. So this is going to be important when you're, again, on inanimate surfaces where you're thinking of trying to just bring the numbers down enough and sanitizing enough that it's not going to have harmful numbers or harmful agents that would be there. So does that make sense? And how we're using those terms? Does it? So in a hospital, do we need everything to be sterilized because people are sick and we don't want to give them something else, right? So are we sterilizing their rooms and are we using everything sterile, right? No, that wouldn't even make sense, would it? Would it even be possible to do it? Mm-hmm. No, but how about an OR? What would be the goal of an OR? What, as far as like where you'd want to be in an OR? Yes, yes, you really do really want that. Yes.
I was watching that video afraid in this boat and like people got bugs, so she like peed on her hand or peed on her hand, like put it all over her body.
She was being eaten up by bugs, and so she wanted to, well, you know, urine is a sterile body fluid. It's supposed to be a sterile body fluid. But obviously, it's got some toxins in it too that will cleanse from your system. So, if I were being eaten up by bugs, I'd probably do the same thing with you. If it might help, it might. I don't know what's the kind of the half of it, but it might. And it's usually pretty acidic. So, it's not as acidic as sometimes your skin can be. A healthy urine does have an acidity to it, though. You would want your urine pH to be kind of lower. So could it maybe act as that? I'd probably give it a try too if I was being eaten up by bugs. I don't know. I don't know the answer to why. It probably does help. So anyway, when we think about microbial control methods, does it make sense to you too that there's different types? There's physical, chemical, and then even mechanical ways we can remove microbes or try to reduce their numbers. So the physical, maybe it makes sense to you. The physical, when we think about physical, we're looking at things like heat or radiation. When we are looking at chemical, we're thinking about gases and sometimes chemical liquids. And when we're looking at mechanical removal, you're usually thinking about filtration. So you're filtering air. You all know about HEPA filters, don't you? Or they're helping you clean air. We'll talk about that in a little bit. But anyway, the whole goals of these, though, look at the goals of some of these. What are they? What are look at it when you're looking at goals? What are the words that are coming up to you? Disinfect. So different kind of goals, right? So sterilization, disinfection, maybe sterilization, disinfection, decontamination. So you're trying to decontaminate. But here you got something on animate. So living versus inanimate objects called antisepsis. And so when you think about antisepsis, you really are thinking about things that can be applied relatively safely to body surfaces. So if somebody's going to have surgery, obviously you're going to have to be more aggressive with the antisepsis than you would if somebody were having just a phlebotomy draw, right? All right, so pretty much this is most of what's in this chapter, what we just did, but there'll be a few other definitions, but I don't think any of that was too tough, right? But here's what I want you guys to know. Let me tell you what we're going to do. I'm going to finish this out because this chapter is kind of quick, but because it's mostly like this. I'm going to finish this out, and then I don't want to give you all anything over the break. I think people need a break. So what I'm going to do, and I'm not going to give the AMP2 people assignments over the break either. So it's not like you come back in on that Monday after your spring break and have to have a test on the last set of information. But what I am going to ask you to do is to sort of think about it, and maybe on that Wednesday, we would have multiple choice tests on this last set of information that we've had since the first test. Multiple choice would probably be that Wednesday or maybe even the Monday after. But let me say this. Chapter 10 is on is on chemotherapeutics. So chapter 10 is on antibiotics, antifungals, antiviral agents. That's chapter 10. And let me tell you what I'm going to do with that. And I won't do it until after break. I'm going to give you all a take-home test. That will count as a test for chapter 10. And every single thing will be right out of your book. Fair enough. And that will count as a test. That's for chapter 10. Is that good? I've always done chapter 10 that way because as we're talking about all of this, it's almost impossible for you to learn the names of these antimicrobials, all of them, and be picky about it. I'm not going to do that, but what I'm going to do on this test, everybody's happy now. I know people are waiting out there. But what I'm going to do on this test is to just make sure that you're getting to hear their names sung so that you do know there's differences between antibiotics versus antivirals versus antifungals versus antiprotozoals. Is that good? Is that fair? So that's going to be after the break though. So actually for today, I don't really have anything for you all. I'm not going to put up, I'll put the lecture recording, if it makes any sense. Anyway, I'll put that up over the guys. I might listen to it myself just to see how crazy I sound. So anyway, I'm going to put that up, but there's not going to be a study guide review today. There will be no lecture quiz Wednesday. There will be no lecture quiz Wednesday. Well, what I'd like you to do is just look at this terminology from chapter 9 and be kind of familiar with it, just a little bit of it. Am I going to ask you any picky details in chapter 9 about a chemical and like what it would best be used for? No, because whenever you're working in a hospital, I know I have nurses in here that are working. You all, it's protocol what you have to use for certain cleanups. So you don't really get to decide. It's going to be protocol for you, but I just want you to know that this is where those infection control officers that you have in your facilities, where they're getting this information. They go to the charts, they look at the chemicals, they see where the chemicals are going to be the most effective, you know, for the whatever reasons, and we'll talk about some of the reasons they choose what they choose, but you're not going to have to know that picky detail. Fair enough. All right. Yes.
I have something on.
Is that the deer thing? Yeah, I want to see it.
It's from the CDC.
From the CDC?
Yes.
All right.